So my work focuses on understanding the evolutionary consequences of hybridization. Okay. My work focuses on understanding the evolutionary consequences of hybridization. And this has been a really interesting area to study over the past few years because we're realizing that more and more species hybridize. And this is in part because we now can see with genomic techniques. Better?
Yes.
We now can see with genomic techniques ancient hybridization events or historical hybridization events that were previously invisible to us. And so over the past few years, there's been a lot of work showing what pairs of species hybridize, what proportion of their genomes are derived from hybridization. And I think that this is really interesting and important work. But I feel like our next step with all of this data is to begin to understand the sort of evolutionary consequences. So how do selection and demography together interact to shape the hybrid ancestry that we see in modern genomes? So as a part of that, I think there are several questions that we can begin to answer in a lot of the data that we already have. And those are hybridization-derived regions randomly distributed throughout the genome, or do we see that there is constraint in where they can occur? Another question is, when we see hybrid ancestry in the genome, is it largely stabilized, or is it still segregating at low frequencies? And finally, in regions that are fixed and hybridization-derived, do we think that these have been fixed due to genetic drift over very long time periods, or do we think that these have been driven to stabilization by selection? So I work on a group of species, the swordtail fish, that hybridize commonly. And they're a great example to answer some of these questions because they have both historical hybridization events that occur between species that no longer overlap in ranges and accident hybridization events between young, closely related species. So I'm going to tell you about one of these historical hybridization events that we inferred based on RNA-seq data occurred between Xiphophorus nezi. It's actually Nezawakatl, but I will trip over my words if I say that too many times during this talk. So here in green, and Cortezi here in red. And what I'm showing you here in this phylogeny is a maximum likelihood phylogeny for the entire genome. And so based on that, we can infer that Montezuma and nezi are sister species, and Cortezi is an outgroup to the clade. And this is exactly the kind of situation where we can easily detect hybridization between nezi and Cortezi, because if we're looking at a particular region of the genome that's hybridization derived, the phylogeny will be different in that region. So in order to do this project, we collected whole genome sequences from 4 individuals. And I've shown you the range of these species here, and you can see that nezi and Cortezi share a border here. But we do think this is a historical hybridization event because that river that separates them is pretty wide and rapid and difficult for a small fish to cross. So for this project, we collected samples from the starred locations and sequenced about 30x coverage. And basically what I want you to notice is that we have 2 samples from nezi. So if we have any introgression from Cortezi into nezi, we can start to answer some of those questions that I posed earlier. Okay, so for this project, we used several different methods to characterize hybridization across the genome. And they were all pretty much concordant with each other, but I'm going to focus on this one that we hadn't used before and we really liked called PhyloNet HMS. And this is a phylogenetic hidden Markov model that dynamically switches between topologies as it goes along the genome. And so in our case, it is switching between the species tree where Montezuma and nezi are most closely related, and the hybridization tree where nezi and Cortezi are most closely related. And the catch here is this method is intended to distinguish between hybridization and incomplete lineage sorting. And the way that it does this is it embedded within the species tree and hybridization tree has all possible gene tree topologies. And we found that it worked pretty well for our data, and I'm happy to talk more about the method if people are interested. So here I'm showing you a simulation result. And this is basically just a single simulation, but I'll summarize them for you in a second, where I've inserted a region of hybrid ancestry into an 80 kb region. And you can see as the method slides along the contig, the posterior probability of hybrid ancestry rapidly switches around this region. And not only does it detect the region, it also infers the boundaries quite well. So that is a nice property. So this is just a single simulation. but overall we found that this method was sensitive in a broad range of parameter space to regions that were hybridization derived that were greater than 10 kb in length. And given the results of our data, we think that we have about a 2% false discovery rate in our data set. And that's likely due to false switches to the hybridization tree due to incomplete lineage sorting. Okay, so I'm showing you here just the raw results for linkage group 1. And you can see that there's quite a lot of switches back and forth between the hybridization tree and the species tree. But if you zoom in on most of these regions, they actually look quite similar to our simulations where you have these sort of discrete regions that support the hybridization tree. So overall, just to summarize what we found from running this analysis, 84% of the genome we were able to assign with high posterior probability, greater than 0.95, to either the hybridization tree or the species tree. And the rest of the genome we think is pretty low information. The reason we actually spent a lot of time looking into this because this method does have some biases in dealing with ancestry heterozygous regions, but we were able to convince ourselves that these are actually just low information regions of the genome. One thing that was quite striking us, even at the beginning of looking at this data, was we did this analysis independently with our 2 NESI genomes, and in both cases we saw that exactly 8% of the genome supported this hybridization tree. So taking a look at these regions just in broad patterns first, we see as expected that these regions have lower divergence between NESI and Cortezi than expected genome-wide. But interestingly, they don't have lower divergence between between Cortezi and Montezuma. And that suggests that the predominant direction of introgression was from Cortezi into NESI, because if it were predominantly in the opposite direction, we would actually expect that there would be lower divergence between Montezuma and Cortezi in these regions. So that's kind of a broad scale pattern, but we could actually use a combination of simulations and explicit tests to look at this. So using simulations and the defoil tests that were developed by Peace and Hahn, we pretty much conclude that about 75% of the regions introgress in the direction from Cortezi into NESI. So for the rest of the talk, I'm only going to focus on the patterns that are observed in these regions that introgress into NESI. But if you do analyze the data as a whole, the patterns are the same. Okay, so one of the first things that we see that is really striking is that the history of hybrid ancestry is the same in both of these samples. So one of the NESI samples is plotted in blue and the other in red. And you can see that their ancestry, their hybrid ancestry perfectly co-localizes along the chromosome. And that's pretty surprising and tells us a lot about their history. So we're inferring that hybridization occurred here at the southern border of NESI's range. And if this population was still segregating for hybrid ancestry when it migrated northward, we would expect that there wouldn't be such perfect concordance between these samples. So this allowed us to infer that there was some stabilization that occurred following hybridization at this southern range border. And we don't know in absolute terms yet how long it's been since the hybridization event, but we can use the length of the Cortezi haplotypes in NESI to say how long it took for these regions to fix in NESI with some caveats. And we're estimating that fixation occurred fairly rapidly, perhaps only 2,500 generations after hybridization. Okay, so now I'm going to talk about the distribution of these regions and patterns of diversity and divergence in them. And for many of these comparisons, I'm going to be looking at comparisons between Montezuma and Cortezi. And the reason for that is that this is a proxy for divergence between NESI and Cortezi at the time of hybridization. So one of the things that we see that's really obvious is that regions that are hybridization derived, shown by the point there, are extremely divergent compared to the rest of the genome. And to me, this sort of raised a hypothesis that these regions are not very important. They're rapidly evolving between species, and thus maybe there's tolerance of hybrid ancestry in these regions. But interestingly, we see that actually there isn't a difference in the proportion of coding base pairs in these hybridization-derived regions. However, just because something is coding doesn't mean it's really functionally important. So if we look at patterns, this is still showing the same divergence plot I showed you earlier. If we look at patterns within these regions of non-synonymous and synonymous substitutions, we see an excess of non-synonymous substitutions, and even in some analyses, an excess in the rate of non-synonymous substitution. So this suggested to us that there may be a lack of constraint on genes in these regions. So in order to test that explicitly, we used the FastCons pipeline from the CEPA lab to identify constrained elements in swordtails. And to do that, we aligned a bunch of high-quality fish genomes and basically looked for evidence of conserved base pairs. So we were able to find 400,000 highly conserved elements in swordtails, and each of these elements comes with its own conservation score, which reflects how strongly constrained it has been across fish evolution. And I'll come back to that in a minute. So the really striking thing that we see looking at this data is that regions that are hybridization derived have about a 15% deficit in highly conserved base pairs compared to the rest of the genome, which we are interpreting as perhaps evidence that there's constraint in where hybrid ancestry can occur in the genome. That in these most highly conserved, most evolutionary important base pairs, we see very little hybrid ancestry, even controlling for power issues. And interestingly, when we even look at the distribution of conserved base pairs, so all of these are highly conserved base pairs, but we say which of these are least highly conserved, which are most. We see that in the most highly conserved base pairs, we have the lowest probability of those occurring in a region of hybrid ancestry. Okay, I think I went pretty fast. But overall, we see that hybridization occurred thousands of generations ago in Nezi and Cortezi. We see evidence that these regions derived from hybridization are functionally distinct and that the genome has stabilized. And finally, we think that the distribution of these regions and patterns of diversity and divergence suggests that there's a possible role for selection in constraining where hybrid ancestry occurs in the genome. However, clearly demography plays an important role, which we didn't explicitly look at in this project. Okay, I want to thank my mentors and people who helped on this project and take any questions. So we did do some Go analyses of these regions, and there were lots of significantly enriched categories, but then I also did some null simulations that also had a lot of significantly enriched categories, and the true hybridization derived regions were not more, they didn't have more categories or more more highly significant categories.
These regions of constrained hybridization, do you think, would you like to speculate on what mechanism might be involved? Do you think it's maybe static interactions or?
Yeah, I mean, I think epistasis plays a huge role. And we didn't, because everything was fixed, we weren't really able to scan for epistatic interactions. One thing we haven't done with this data set that I've been meaning to do is like a pathway type analysis. We did do some sort of basic pathway analysis, but we didn't look at, try to look at particular, we didn't do it in such a fine-grained way that I think we've excluded that. So that would be one way to get at that.
A related question, how do you think this constraint to ancestry will scale with divergence time between the lineages hybridizing?
Yeah, so these species, I mean, this was,